Hello, today we will be studying about chlamydia. Now, let us see the history of chlamydia. There was the first outbreak of this organism in the US, that was in 1929. Then you can see the first cultured. It was not able to be cultured then when it was studied. Then it was first cultured in the year 1956. You can just see outbreak was in the year 1929, but it was able to be cultured only in the year 1956 by Tang Fu Fan. It was not understood as a bacteria initially because bacteria is a free living organism. This organism requires a host, and hence it was not understood as a bacteria. But later on, it was understood, recognized as a bacteria in the year 1966. Now, I had told you that all the organisms, all the bacteria that has been identified till now and studied is being placed in Bergey's manual. Okay, so even this organism has to be placed in the Bergey's manual if it is a bacteria. So there was, as I had told you initially, they were not able to know whether it is a bacteria or not. Later on, it got its place in the Bergey's manual between the year 1989 to 1999. So this is about the history. Coming to the classification, it belongs to domain bacteria, phylum Chlamydiae, order Chlamydiales, family Chlamydiaceae, and genus Chlamydia. Now, what are the characters features of this organism? If you see, it is a bacteria, so obviously it has to be either gram negative or gram positive. It is a gram negative bacteria. The shape is either oval to spherical. The size ranges from 0.2 to 1 micrometer. Initially, it was believed that it does not have a peptidoglycan, but later on, the recent studies have shown that it has a peptidoglycan and also associated other proteins with it. As I already told you, it is a obligate intracellular parasite, which means it requires a host always for its survival and multiplication. It cannot survive outside, it's not free living, which means it requires a eukaryotic cell. It can survive always inside a eukaryotic cell only, and inside vesicles only it will survive. It also multiplies inside the host. You can see in this picture here, this is the entire eukaryotic cell and this is the organism where it is present. Okay, in this picture also you can see this is a eukaryotic cell and there are these small things which is shown. Okay, small dots which is shown as the bacteria Chlamydia. Now it is susceptible to most of the antimicrobial agents. I am not saying antibiotics, but to most of the antimicrobial agents. Now, why it is not able to survive freely? Because it is not able to produce or generate its own ATP. Uh, it has to survive, or it has to depend on the host for the ATP. So that is why it requires a host. In other words, it depends on the host ATP for its energy. Coming to the media, as this organism cannot survive freely, hence definitely it cannot be cultured in a bacterial media. It requires to be grown in some living tissue or embryonical eggs. Coming to its life cycle, now if we have to study about its life cycle, it is basically present in two forms. Okay, now its morphology or its cell, it is present in two forms. Remember this: elementary bodies, or it is called as EB, elementary bodies, and reticulate bodies, or RB. Remember, in the exam they can ask you for 1 or 2 marks EB, so that's elementary bodies and reticulate bodies. Elementary bodies are those which is outside the cell and they are inactive form. Okay, reticulate bodies are those when the organism is inside the host. So that is called as reticulate bodies. So remember this. Okay, now coming to the life cycle. Now I told you that the elementary bodies are those which is present outside the cell. So they are inactive form. So these are the elementary bodies which is seen in green here. Okay, now these elementary bodies, as long as it is outside, it is as elementary bodies. Now it attaches itself to the host, to the cell, you can see here, and it enters inside the cell through phagocytosis or through phagosome. After that, it gets converted into RB. What is RB? Reticulate bodies. So reticulate bodies forms a sheath around itself. You can see a sheath here that is formed. Around itself a sheath is formed. Okay, and inside is the reticulate body. So a sheath is produced. Then once the sheath is generated, the reticulate bodies now starts multiplying. You can see there was only one reticulate body, but now there are four reticulate bodies. It gets multiplied. Many reticulate bodies are formed inside the sheath, you can see here. Okay, now once the it is time for the reticulate bodies to come up, to come out. Okay, then what happens is the reticulate bodies gets converted now to the elementary bodies, because it has to come out, and the if it is present outside, I already told you, it is called as elementary bodies. Then slowly, once it gets converted to elementary bodies, that sheath gets dissolved, and then the the membrane, the wall of the eukaryotic cell in which the elementary bodies or the reticulate bodies were present, is bursted open, and it comes out, the organism comes out as elementary bodies. Again the cycle continues. So in the exam you are expected to draw this cycle as such. Coming to the important species, what are the different species? There are many different species, but the important ones are Chlamydia trachomatis, Chlamydia pneumoniae, and Chlamydia psittaci. The first one, Chlamydia trachomatis. Now what does it cause? It causes trachoma. Okay, now what is this trachoma? It is the infection of the eye. It's also called as a pink eye disease, Egyptian pink eye disease, or it is also called as Egyptian ophthalmia. Okay, so these are the different names. It's the same thing. It is a trachoma. What are the symptoms? As the name tells, pink eye disease, the eyes become pink, red to pink. Okay, the inner eyelids becomes rough. There is pain and breakdown of the outer surface of the corona, and ultimately it can lead to blindness. Now how does it spread? Obviously poor sanitation. When there is too much of crowd, it spreads. It is through direct or indirect contact. That is, the person who has been infected with this rubs the eye, and you give a handshake and you rub your eye. So that's how the organism transfers. Indirect contact, that is through fomites. You can say the through clothes, through inanimate objects, that is stable. The person who is infected will rub the eye and touch some inanimate object, say cloth, and you touch the cloth and then you rub your eye. So that's how it get transfers. Okay, it's more common in children and in women. Okay, children because the immunity is less. Women, why? Because the women are always in close contact with the children, so that's how it's more in women. Incubation period is 6 to 12 days, and the treatment is either by medication or surgery. Surgery, removal of that part. Medication, either you take azithromycin or you take tetracycline. Coming to the next one, Chlamydia pneumoniae. As the name itself tells, pneumonia, it causes pneumonia. Okay, it was called as TWAR, that is Taiwan acute respiratory agent, because it causes infection in the respiratory tract, your upper respiratory tract and the lower respiratory tract. Initially, the organ was named as Chlamydophila pneumoniae, and later on, when they found the organism was nothing but chlamydia, they have named it as Chlamydia pneumoniae. Initially, it was Chlamydophila pneumoniae. The host, generally it was supposed to be frogs, turtles, okay, and there was no human infection seen before 1950, but after that it was seen that humans were also getting infected by this organism. What are the symptoms? You can see pharyngitis, bronchitis, atypical pneumonia. I told you what was the meaning of atypical pneumonia. Sometimes you get pneumonia like symptoms, and then automatically after few days the symptoms are vanished. I mean, you get cured out of that disease. So in the upper respiratory tract, you may have coughing, you may have breathlessness, and here you can see the organism present in the mouth. Coming to the next organism, that is Chlamydia psittaci, it causes psittacosis. Psittacosis is basically infection in the birds, you can say. It is a zoonotic infection, so the infection can spread from birds to humans. It was seen in parrots, pigeons, and sparrows, um, and this was the first thing that was seen in US. How you had seen in history that it was the first disease that was seen spread through the organism Chlamydia, because of the pets that were kept, that is pigeons and parrots were kept as pets in the houses of people in the US. Later on, people started getting the disease psittacosis, and later on they started understanding that it was because of the parrots and pigeons. Okay, it also causes atypical pneumonia. Pneumonia like symptoms it has. Apart from pneumonia like symptoms, you also can get joint pains and diarrhea, conjunctivitis, and there is low level of WBC. Now, why there is low level of WBC? Actually, when there is infection, the amount of WBC increases, but here there is low level of WBC because the host here is, I mean, the organism wants, I told you, organism always survives inside a host, isn't it? So Chlamydia psittaci will survive inside WBC cells. So every time it enters a WBC cell, the WBC cell is lysed when it comes out. Incubation is 5 to 19 days. Route of infection, how does it spread? Through the droppings of the bird. Okay. Otherwise, also through the respiratory secretions, through aerosols. Treatment would be tetracycline and chloramphenicol. Here you can see the diagram of the organism. So these are the different three main important species of chlamydia. Thank you.